Okay, so good evening, everybody. As you all might know, I am Sanket from IIT Delhi. So, as few of you pointed out, that why is sheltering required to prevent, to make DNA repair enzymes inaccessible to telomeres. It is important because telomeres in itself, if they are close together, then they resemble a double-stranded break. So essentially, if shelterins are not available, the repair enzymes will consider these as double-stranded breaks and will repair them. So two chromosomes will be joined together. So that's the idea. And then coming to the DNA damage by Ross, so we have already discussed that guanine is converted to 8-oxoguanine, right? That we have studied earlier. Some other damages that happen due to reactive oxygen species is the conversion of thymine to thymine glycol. Something else also happens. Uracil gets converted to 8-hydroxyuracil. This also happens. So these are some modified bases that are present in our DNA. They are the signatures of reactive oxygen species. So how would we measure the extent of reactive oxygen species damage? Could anybody tell? Anyone? Maybe raise of hands. No one. Okay, Antik.
Sir, we might measure the absorbance maybe at
Not
260 nanometer.
don't, no, don't tell me about absorbance and these kind of things. Be more simple. As I said, that guanine is converted to 8-oxoguanine, thymine is converted to thymine glycol due to reactive oxygen species. So what do we need to measure? Thymine glycol and 8-oxoguanosine, right? If we can measure these things, we can know that what is the extent of uh, ROS, I mean reactive oxygen species induced damage. Is that clear?
Yes, sir.
Okay, so exactly this thing was done in this paper by Bruce Ames. So they wanted to assess what was the oxidative damage to DNA, right? So essentially, what they measured were the amount of thymine glycol and thymidine glycol. Okay, in the urine output. So essentially, what happens if your body has these bases, right? Thymine glycol, thymidine glycol, these kind of bases are present. The DNA repair machinery will, remove these. They will remove these, and these will be finally coming out in your urine, in your urinary output. So that was the essential idea behind this. So for thymine glycol, there is around like 300 hits per day. I mean, I'm just telling you, this was not discussed in the paper as such, but 300 hits per day of like thymine glycol production is there. Similarly, for hydroxymethyl uracil, another thing which we have discussed, there is around 700 hits per day. So, I mean, the extent of this damage is very clear. So, if there are 300 hits per day or 700 hits per day, all of these are not they do not stay as such in your DNA. If so happens, then what will happen? Your DNA will be damaged. So your DNA repair machinery is efficient enough to remove most of the thymine glycol and thymidine glycol. So let's see what happens. Essentially, they had taken 4 mice and 3 monkeys. So the amount of thymine glycol and thymidine glycol, as you can see here, is given. So what is the observation that you have? Anybody can tell that. What is the
The
observation?
average, the average
Huh?
for thymidine glycol is much less than as compared to thymine glycol.
That is fine. Okay, good one.
So,
Good observation. Other than that, between monkey and mouse.
Between monkey and mouse, it is evident that oxygen consumption is more in case of no wait, but I'm telling.
Not this one. I'll ask about this also, but what happens here? I'm asking here. Can you see the annotations? I hope you can. Yeah, please go ahead.
The amount of mutations or the amount of oxidative damage in mouse is more than that of monkey. Extent of oxidative damage is more.
Is more okay. Sorry for the interruption. Thymine glycol is more in the urinary output of a mouse than that of a monkey, right? Am I correct?
Yes, sir.
Okay. So your inference that the DNA damage is more is true actually, but right from here we cannot do that because, as I said, the the output, the urinary output is dependent upon the DNA repair mechanism. So we can also infer that the DNA repair mechanism of mouse is better than that of monkey. Can we say that? Maybe it is possible. Right from this data, it is possible, but we cannot infer that right now. In the right hand side, what we see, there is oxygen consumption in the x-axis, urinary output of thymine glycol and thymidine glycol in the y-axis. So what do we see here, Antik? What is your observation?
For the same oxygen consumption, the urinary output in that first graph is more. Wait. Sorry. For the same oxygen consumption, the amount of thymine glycol formed is more as compared to thymidine glycol.
Okay, that is a good observation. Then
So now, with respect to that, that means that if we, I don't know whether I'm correct or not, but the extent of like those straight lines which are showing that the extent of thymine glycol formation or the thymine glycol urinary output is a bit more in case of rat maybe as compared to mouse. I don't know whether it is.
okay, these are called correlation plots. So essentially, if you see a mouse has a higher oxygen consumption, can I say that?
Yes, sir.
And it has higher thymine glycol urinary output. That I can say.
Yes, sir.
For a man, the oxygen consumption is lower, and the thymine glycol production is also lower. I mean, in urine, right?
Yes, sir.
So can I say from this data that the metabolic activity, so metabolic activity is essentially the amount of oxygen consumed, right? Metabolic activity is kind of inversely proportional to the no, it's not inversely proportional. It's directly proportional to the thymine glycol or thymidine glycol production. Is that correct? I mean, are you guys following this?
Yes, sir.
Yes, sir.
Okay. Another thing, how is a mouse different from a man? I mean, I mean, this is another observation which you should have actually. So mouse is, how would you like say, how is it different?
The metabolic activity
That I said,
is.
you see a mouse, you see
And
a human. What differences do you expect? Anything?
structurally different. Sorry.
Do you see a mouse that is of human size? No, right? No. So mouse is of the least size, right? Among mammals, one of the like least size is of that of a mouse. Humans are, on the other hand, higher order animals. They have a higher size, I mean, more size. So, can you see a correlation between the size of an organism and the metabolic activity?
Yes, sir.
These are the observations essentially from here. So, the same thing, that higher oxygen consumption translates to higher metabolic activity, that I have already said. So for a man, the metabolic activity is lower than that of a mouse. So this is the order essentially. So higher metabolic rate directly corresponds to increasing urinary output of thymine glycol and thymidine glycol. So all of you agree with this?
Yes, sir.
Okay, so does the lifespan of an organism inversely proportional to the oxygen consumption and metabolism? So let's go back. Let's go back to this slide. So uh, who lives longer, mouse or man?
Mouse. Hey, man. Sorry. Oh,
Anyway, fine. Okay, so we can say, right, it's a preliminary analysis which we can, I mean, it's not an like sufficient data we have to back up this claim, but we can definitely say that lifespan is dependent on the rate of oxygen consumption or metabolism. We can also say that the lifespan is dependent upon the size of the organism. So if you take an example of an elephant, it lives much longer, right, than humans, don't they? So these are very basic principles actually. So through this study, we came to know that DNA damage plays a significant role in the aging process, since lifespan is also dependent upon these organisms. So we say that if an organism suffers more damage due to its higher metabolic rate, then its lifespan is smaller. So the aging process is faster. So larger animals live longer because they have lower specific metabolic rate and hence lower oxidative damage. Why do I mention specific metabolic rate? Why don't I just say that they have a lower metabolic rate? Anyone?
per unit mass. Like
Yes.
on the
Yes.
same basis,
So
it will be.
naturally, if you see that humans have a much larger amount of cells than that of uh, mouse. So if you just compare the metabolic rate, then you will find that humans have a higher metabolic rate. But if you compare it per unit mass, then you will see that mouse, they have a higher specific metabolic rate compared to humans. So specific metabolic rate is important. Now coming to the consequence of DNA damage. Now DNA damage has occurred. What can happen? Two things can happen. Either the DNA damage can persist. It remains in your system. It is fixed as mutation. And then if it is fixed as mutation, there is a higher chance of DNA damage, right? Another thing that can happen is that this increases the mitotic activity of the cell, right? So it will increase the cell division, now frequency of cell division due to this mutation. So if this frequency of cell division increases, then the damage increases, and then again it's a cycle basically. So in which case do you think that there is more number of cell division, more increase in damage, and it goes uncontrolled? What is that disease? Anybody? We just talked about it in the last class. It's cancer, right? Okay. Can you people hear me?
Yes, sir.
Okay.
Sir,
So if
can you
mutation,
just, sir, can you just
Mhm.
repeat all the inferences once more? I couldn't note them down uh, of the previous
I'll give
explanation.
you the slides. No, I think it's fine if you, or do you need me to repeat it? This one, right?
Like the first, like that table thing.
Here.
Yeah, this one. There were two inferences, like from this table. Can you please repeat them? I couldn't note them down.
So essentially, the amount of thymine glycol is directly proportional to the metabolic activity of. Okay, from this table. Okay, so here the mouse, they have a higher thymine glycol production than that of a monkey, right? So there is, mouse, we know they have a higher oxygen consumption. So if there is a higher oxygen consumption, higher metabolic activity, then there is more DNA damage. We'll come back to it. I think we'll come back to a few other papers. You'll understand. I mean, the inference is better. We'll have more like papers. Is that okay with you?
Yes, sir.
Okay. Now, just give me a moment. Let's go back. The consequences of DNA damages were discussed, and then we said if mutations they compromise the integrity of cells, then there will be faster cell division and there will be more persistence of the damage, and the next generation will also get damaged. I mean, get the damage essentially. And DNA repair mechanisms in humans is more efficient than that of mice, that we know, right? And the cellular turnover in mice is higher than that of humans. So, I mean, the cell division in mice is higher than that of humans. So, if the cell division is higher, then if there is a damage that will get divided faster, that will be spread faster. Is it okay? I mean, is it clear to all of you?
Yes, sir.
Okay. So, have you, any one of you have read articles that, animals like elephants, they are more, immune to cancer. They don't get cancer so easily. You have read about it. If, I mean, maybe read about it or something. You people know about this?
No,
No,
sir.
but no. So that's actually, that is something that happens. Please look into it. You will find a lot of interesting articles. I mean, even news articles in such cases. So one of the reasons is they divide very slowly. So now we are seeing this 8-hydroxy guanosine in the kidneys and in the intestine, even in the liver. You and this is the age essentially. The x-axis is age and the y-axis is 8-hydroxy guanosine. The I mean, amount of 8-hydroxy guan. What is the observation? Anyone would like to say? This happens in case of mice. This is a study in mice. Age is increasing in x-axis. This is the increasing concentration of 8-hydroxy guanosine. Why are we studying 8-hydroxy guanosine?
Sir, can you repeat the question?
Why are we studying 8-hydroxy guanosine here? Is the question not clear yet?
Yes, sir, clear. But since it is a mutated base nucleotide, that's the reason. Mutated
I'll
base,
go back to the first slide. I'll go
chemically
back to
modified.
the first slide. Okay. Why are we now studying 8-hydroxy or 8-oxoguanosine? 8-hydroxy guanosine is kind of like similar only. It's also a modified base. So why are we studying it?
To measure the extent of DNA damage by
Due to
reactive
Ross.
oxygen species.
Yes,
Ross.
right. Good. Okay. Is everybody clear with that?
Yes, sir.
Okay. Then what is the observation here on the left hand side, left panel?
With increasing age, the amount of 8-hydroxyguanosine that increases, and this increase is recorded to be highest in the liver, some intermediate value in the kidney, and a bit lower in the intestine. Or kidney and intestine, we cannot differentiate too much. Uh, they're almost similar, but in the brain and testis, the amount of 8-oxo-guanine production, 8-hydroxyguanosine production is almost constant.
Okay, fine. That is okay. So we can say that in the liver tissue, if we only take the example of liver for it now, in the liver tissue with increasing age, there is more incorporation of 8-hydroxyguanosine, 8-oxoguanosine, as whatever you say. So there is more incorporation of that. Now here in the right hand side, this is the urinary output. Okay, urinary output of 8-hydroxyguanosine in picomole per kg per day. So mice, this is also the same study by the mice were studied. So when they were 2 months old, there is more amount of 8-hydroxyguanosine. When they are 12 months old, it is lower. So the trend is like this. Okay, here. So what does this mean? What is the inference here? I mean, what is the observation? Then we will talk about inference. Nobody? Are you guys not understanding?
It is decreasing. Means with age, the urinary output of 8-hydroxyguanosine is decreasing. So
Fine, fine.
it might be, and it is a bit increasing in the last later stage of the life. It is increasing a bit, uh, but the thing is that if we compare this with the previous graphs, like on the left side, so it might be the fact that the amount of 8-hydroxyguanosine that is remaining in the means the genome sequence, that might be persisting, more persisting with age. So problems are arising for this, which is leading to death of that.
Okay, I really could not hear you properly. Probably there is an internet connection failure here. But I'll tell you, I really don't know what you said. Am I audible, by the way?
Yes, sir.
Yes, no. I mean, maybe type it. I can see
Yes, sir.
better. Okay. So essentially what is happening here is, wait, I'll just switch off my video. I think it will be better in that way. Just give me a moment. Okay. So here, essentially, what happens with increasing age, the urinary output of 8-hydroxyguanosine decreases. Okay, that is clear to everybody. Now, why that happens? So the DNA repair machinery is more effective at a younger age. Okay, it's more effective at a younger age. That's why whenever it is encountering an 8-hydroxyguanosine, it is excreting it out through urine. Fine. But when the age is increasing, the DNA repair, efficiency is decreasing. Is that clear?
Yes, sir.
Okay. I hope you said the same thing. So again, lifespan of a species is inversely proportional to its specific metabolic rate. We have talked about it. Yes, it is possible. Now the of altered nucleotide 8-hydroxyguanosine, there is a rate of accumulation of 8-hydroxyguanosine which increases with an increasing age. So usually out of every 55 mutations, 54 get repaired, but one remains permanent. So the DNA damage, extent of DNA damage is still small due to this reactive oxygen species. However, uh, as we said that 8-hydroxyguanosine, in the urinary output, it decreases with age. This might be due to the reduction in the efficiency of the DNA repair mechanisms. So one more thing that we observed was that the larger mammals have a longer lifespan. So you know examples, as I said, elephant, a lot of times. So they have a very long lifespan. So if you take and consider an example of mice now, they have a shorter lifespan. Humans are somewhere in the middle. So they have an average lifespan. So an exception to this is turtle, which there is it's a small animal, but it has a very huge lifespan. So there are various other reasons also apart from the size itself. So essentially, when we talk about the oxidative damage in mitochondria, uh, what happens? Why are mitochondrial DNA more prone to damage? It is because of the lack of protective histones. Are you all, aware of it? Histones are not present in mitochondrial DNA.
Yes, sir.
Okay. Now the DNA repair mechanisms are not that efficient for mitochondrial DNA, and mitochondrial damage mostly happens because of replication errors. Okay, that is one thing. Another thing which is not written here is that the oxidative environment of the mitochondria. Oxidative phosphorylation happens in mitochondria.
Yes, sir.
So oxidative environment, there is higher amount of reactive oxygen species that is present in the mitochondria. So definitely the damage will be higher in the place where it is present, right? So you can see if there is a higher amount of reactive oxygen species and there is less amount of antioxidant defense. So any example of any antioxidant defense mechanism, any enzyme that you know?
Peroxidase, catalase.
Yes, peroxidase, catalase, superoxide dismutase, everything is essentially antioxidant defense, right? So if there is reduction in that, increase in the reactive oxygen species, there will be more mitochondrial damage, fragmentation, mutation, these things will happen. It will cause a malfunctioning mitochondrium. So these are all the errors. These are all the effects of that. And this will lead to an enhanced reactive oxygen species production. And finally, it will cause inflammation, cell injury, apoptosis, and it leads to all these diseases like cardiovascular diseases, chronic kidney diseases, chronic obstructive pulmonary disease, autoimmune disease, mitochondrial myopathies, and neurodegenerative disease, all these kind of things. So mitochondrial damage also increases with age. So the incidence of these kind of diseases also increase when a person is older. Is that clear to everybody?
Yes, sir.
Yes, sir.
Okay, good. Now coming to so this is what we said. So the oxygen free radical will be there. This is essentially ROS. So if there is ROS, then there will be cellular damage, mitochondrial damage, all these kind of things that will increase the process of aging. And in turn, that will cause higher production of ROS and then again cellular damage. So it's a cycle. It's a cycle that continues. So if there is damage, it will increase the damage slowly and steadily later on. So is that clear?
Yes, sir.
Okay, so now we will discuss about the theories essentially. So before I get into this diagram, I would like to give you a brief history. I have like curated. So Max Rubner in 1908, he proposed the rate of metabolism or oxygen consumption and the body mass are related. What I said essentially in a few slides back. So he did that in 1908. He hypothesized this. Then Raymond Pearl in 1928. Pearl, he said that he hypothesized a rate of living hypothesis. So what does it mean? What does he said? So he said that you have a limited energy in your entire lifespan. Okay, so you can only spend a limited amount of energy. Consider it as like a currency. So you have a limited amount of money in your wallet. You can spend only that much amount. If you spend more, you will live less. If you spend less, you will live more. Is that clear?
Yes, sir.
Okay, so how would you relate it to that mice wala experiment, the metabolic rate and everything?
Sir,
Nobody?
repeat. Can you repeat the question? My network is not working.
How would you relate rate of living hypothesis with that experiment by Bruce Ames, where mice had a shorter lifespan, humans have a longer lifespan, and the metabolic rate of mice is higher. I actually gave you the answer.
Yes,
So,
sir.
ah, so anyway, that's fine. So in 1930s, there was another group. So they have studied that hyperthyroidism and hypothyroidism. So in hyperthyroidism, usually the metabolic rate increases and there is more oxygen consumption and there is hyperoxic death. In hypothyroidism, essentially hyperoxic death is delayed, but there are some other complications that come up. So oxygen, I mean, too much of oxygen is also damaging to you, right? So essentially in the 1950s, around at this time, the free radical theory was, introduced, right? So when they kind of found out that free radicals are present essentially. So H2O2, that is hydrogen peroxide, was discovered in the cell, and that was essentially the starting point of free radical theory. Now, then they realized later at around 1970s that mitochondria is involved. So they did not know that how is it produced. So they now realize that mitochondria is involved. So the main important person here, Herman, you will see his name again and again. So he initially proposed the free radical theory, and then he proposed the involvement of mitochondria in free radical theory, and he related that to aging. And once mitochondria was introduced, then the mitochondrial enzymes like the superoxide dismutases were also discovered later. This is the timeline. Now, coming to the basics of Herman's theory, what he said that oxidative stress, it increases with age, right? So the more you age, there will be more amount of ROS produced, and essentially the oxidative stress will increase. So that is, by oxidative stress, I mean that. And there is involvement of mitochondria in this process. Fine. So here also you can see the cartoon here. It is the mitochondria with various functions. So here you can see peroxidases. They, they neutralize the hydrogen peroxide. Few of them, they even go to the cell. So, and these things, you can read about these things later. I mean, we will talk about it in detail. Now let's go to the next slide. So if there is a mitochondrial dysfunction, right? So how can mitochondrial dysfunction happen? It can be due to, any infection. It can be due to some drugs that you're taking, any kind of thing like that, or any chemical, or, UV induced damage. It could be a lot of things. So if there is a dysfunction of mitochondria, there will be an elevated level of reactive oxygen species. Then again, if there is elevated levels of ROS, it will cause metabolic disorders, neurodegenerative diseases, faster aging, cardiac arrest, kidney failure. So everything is related. So mitochondria is a very important, uh, you know, uh, what would I say? It's an element that is very important in your body. So what is mitochondria generally known as? You must have heard it from class 9 or something.
Powerhouse of the
Powerhouse of
cell.
Yeah, that's correct. So if the powerhouse of the cell is not functional, definitely there will be a lot of issues, right? So we will discuss about it in detail. So essentially, when we are talking about electron transfer, right? So essentially in mitochondria, in the previous slides also, you have seen that there is, this oxygen, I mean, peroxide oxygen radicals, they are all created due to, oxidation and reduction reactions. So that happened due to transfer of electrons, right? So redox reaction happens due to transfer of electrons. So what could be the mechanisms of electron transfer? One is direct transfer. So in direct transfer, what happens if, like, this is one oxidized state of iron, it will be convert it to Fe2 plus, and here what it will be there? The electron will be here or here? In the left hand side or the right hand side?
Left hand side.
Okay. Okay, that's correct. Then if there is a transfer of hydrogen atom, so if there is, hydrogen is transferred from, from some membrane, if hydrogen goes here, let's say hydrogen is transferred, then here there will be an electron. Essentially, there will be a charge gradient.
Yes, sir.
You must have read about this in electronics somewhat, a little
Yes,
bit.
sir.
And then there is hydride ions. So hydride ions have 2 electrons instead of 1. So it is having a negative charge. So they can also be transferred. These are the mechanisms of electron transfer. So coming to the TCA cycle, where, after which there is a transfer of electrons at a lot of places. So you see NADH and FADH2, they contain electrons that are relayed to the electron transfer chain. Is that clear? This is the TCA cycle. You are aware of this?
Yes, sir.
Read about it maybe? Do you know?
Yes, sir.
Yes, sir.
Okay, so please focus on these two enzymes, aconitase and succinic dehydrogenase or succinate dehydrogenase. So here this Q is essentially coenzyme Q. Just remember this, we'll come back to this again. Coenzyme Q, so it is also called ubiquinone. Fine. It's also called ubiquinone, which is converted to semiquinone. As you can see here, the QH2 is semi-ubiquinone. So this is essentially, they are important in electron transfer chain. So we will discuss how they are important. So this is in a mitochondrial inner membrane. This is complex 1, complex 2, 3, 4, and this is the ATP synthase. Are you all aware of these?
Yes, sir.
Yes, sir.
Okay, so essentially what happens if I enlarge this image, so NADH, it gets converted to NAD plus, right? So there is transfer of electrons. Whenever there is transfer of electrons, you will observe at each and every, complex except this complex 2, there is pumping of, hydrogen ions outside the mitochondrial matrix. Is it, I mean, are you guys aware of this?
Yes, sir.
And this same proton gradient, which is called the proton motive force, is used to synthesize ATP. So that is how a mitochondria synthesizes ATP. ATP is the, the currency of energetics. In terms of energetics, the currency of the cell, in a way. So that is how it is produced. So you people are aware of it, right?
Yes, sir.
Okay. So coming to each and every complex. So you see, so the complex 1, if you see, they are clusters of iron and sulfur. Fine. These are clusters of iron and sulfur, and which are important for relaying of electrons, right? They're important for relaying of electrons. So sulfur, not sulfur, iron can exist in stages. So Fe3 plus and Fe2 plus. So you have already seen how direct transfer happens. Is it clear?
Yes, sir.
So the same reaction will happen, and here there will be NADH, which will have, um, I mean, NADH, NAD plus H plus. So here the electron will be transferred to this FES cluster, and this will then go to this complex where Q is again ubiquinone, which will be converted into semi-ubiquinone, and during this process, hydrogen will be pumped out. of the mitochondrial matrix, right? Is that clear? There are two sites, this Ty, Ky and PSST. At these two sites, this relaying of electrons happen and ubiquinone is converted to semi-ubiquinone. Is that clear? Or is there some
Sir, repeat
confusion?
sir, please repeat that last part which you said.
See, yahan se electron is coming, it's getting transferred. Here also there is electron relaying of electrons. So Q minus se QH2 ho raha hai, so electron is relayed. So in this process, what is happening? Hydrogen ions are pumped out to transfer these electrons. So this happened at two sites in the inner mitochondrial membrane. These are the two sites, and these are the inhibitors. So if you use rotenone, okay, especially rotenone, if you use it, then it has a very it makes this Q, I mean ubiquinone, very stable. It does not get in form semi-ubiquinone. So what would happen to the electron? Electron will not be relayed, right?
Yes, sir.
So what will this electron do?
It will stay there, like
Stay there.
whatever.
What it will do? If there are oxygen in this cell, I mean in the mitochondrial place, if there is oxygen, then what will happen?
Yeah. That will
Sir, can
react.
you repeat the question?
Electron is not relayed because you use rotenone and the Q is not getting converted to Q minus. Fine. This is an inhibitor of this process. This yahan ka process. Fine. Now, if it is not relayed, if oxygen, I mean electron is not relayed, then what will happen? Electron will be left out.
Yeah, oxygen concentration will increase.
What will increase?
Oxygen concentration will increase.
What will happen in this reaction? Tell me.
Which one?
Can you not see this? I mean, I've annotated oxygen, O2 plus electron. What will happen?
A free radical, like superoxygen, I guess.
Superoxide radical.
Yeah,
What is this? A reactive oxygen species.
yeah, it will inhibit. Yeah,
So essentially that what happens, that is what happens. So oxygen gets converted into a superoxide. So if we use rotenone, what would you expect in a cell? Enhanced reactive oxygen species, right? So
Yes, sir.
similarly, this is cytochrome. I mean, sorry, complex 3. Okay, it has complex 3. It accepts the electrons generated in complex 1 and complex 2, and they relay the electron from cytochrome B to C1C to complex 4. So it will give the electrons to complex 4. Here also you will see that there are this same ubiquinone, semi-ubiquinone, these are present, which relay the oxygen. And how do you understand that the oxygen is relayed? Because hydrogen is pumped out. So if we go back to this slide, I mean, let's go back to this slide. So which kind of transfer is it? I mean, first, second, or third? Transfer of an hydrogen atom, right? That is what it
Yes,
is.
sir.
So that means if hydrogen is going out, electrons are coming in. That is the idea, right? So यहाँ पे electron is not specifically mentioned. I mean, electrons are not shown as such. But when hydrogen ions are removed, then we will assume that electrons are coming in, right?
Yes, sir.
Okay, that is what it is. Then this is the entire schematic here. Okay, so this is ubiquinone to semi-ubiquinone. I mean, sorry, this is not that ubiquinone complex UQ10. It has an electron. Now it is transferring its electron to the oxygen radical. So there will be a production of superoxide radical, right? Superoxide radical is produced. Now in this process, this superoxide radical is neutralized to hydrogen peroxide using the enzyme superoxide dismutase. Is that clear?
Yes, sir.
Now this hydrogen peroxide which is produced, some of you said about peroxidases and catalases. So this catalase and peroxidase, they will neutralize it into water. So it will be essentially non-oxidative. There will be no damage to it. Fine. Is that clear? Is it okay? I mean, is it clear? Please show your hands if it is clear, or I will explain it again. Okay. There's only one hand. What is not?
So, is the entire process like a chain, or is it a step-by-step process? That's what we are not understanding.
It happens, I mean, one after other, right? That it's not like I mean, superoxide radicals are produced and then they are neutralized as well. I mean, there are enzymes to neutralize them as well in mitochondria. So the same thing happens. But if let's go forward, if there is a more amount of superoxide produced, then what will happen? Aconitase and succinate dehydrogenase. Do you remember this slide? It's not going to this slide. So this slide, wait, let me go back to the slide of TCA cycle. So aconitase and succinic dehydrogenase, they also have FES clusters, like we have in the complexes of mitochondria. They also have FES clusters. So these FES clusters are attacked by the superoxide radical. So when they are attacked, it forms this aconitase, which is now non-functional. If it is 3Fe4S, it is non-functional. So generation of reactive oxygen species will also have an effect in the TCA cycle. Is that clear?
Yes, sir.
Now this jo Fe2 plus is released because of this process. If Fe2 plus is released, it will convert the peroxide into OH radicals. This OH radical is even more damaging than hydrogen peroxide and superoxide radical. Fine.
Yes, sir.
So this is what happens essentially in a cell. So to combat the superoxide, essentially to combat the superoxide so that it doesn't stay for long in a mitochondria, there are enzymes, there are cytosolic intramitochondrial superoxide dismutases and extracellular superoxide dismutases. So the cytosolic and extracellular superoxide dismutases, they have CuZD, like they have copper and zinc in their protein structure. They are SOD1 and SOD3 respectively. I mean, they are encoded by these genes. And intramitochondrial superoxide dismutases, it has MNSOD and it is encoded by the SOD2 gene. Now, why is this important? We'll come back to it. Later, so here we see the same thing, how superoxide dismutases are present. So you see in the cytosol, there is CuZnSOD, which converts superoxide radical to hydrogen peroxide. Hydrogen peroxide using peroxidase enzyme is converted into water. But what is this reaction then? What is this reaction?
And
Penton reaction, right? So if there is more amount of hydrogen peroxide, then there will be a production of hydroxyl radical. Now this hydroxyl radical, it is very damaging to the cell. It also damages the telomere apparently. So there is telomere, enhanced telomere shortening. So if there is enhanced telomere shortening, what would happen? Do you remember
Sudden
the
death.
hallmarks? Hallmarks of aging?
Proteostasis.
No, 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 not that. Telomere attrition was a hallmark of aging. You remember?
Yes, sir.
So that is exactly what is happening. So there will be faster aging. So the question arises that reactive oxygen species is more produced in larger quantities due to age, or reactive oxygen species causes faster aging. That is a question that a lot of people cannot answer. It's like a chicken and egg problem. We really don't know. But what we know is when there is, reactive oxygen species, they cause damage. This damage causes a faster aging, and then aging in itself causes enhanced production of ROS. So it's kind of a cycle, right? Is that clear?
Yes, sir.
Yes, sir.
Okay, then if in the mitochondria, this is the mitochondrial matrix. Okay, mitochondrial matrix. So here you will observe that this superoxide radical is converted to hydrogen peroxide using MnSOD. So then it is neutralized using the peroxidase enzyme. And same if here the Fenton reaction happens, then this hydroxyl radical will damage the mitochondrial DNA. So you know the mitochondrial DNA, why it is damaged right now? I mean, is it clear to all of you?
Yes, sir.
Okay, then next, let's go little bit. I mean, we will come back to this slide once again in a later part of our course, but since I think it is somehow relevant here. So here we are seeing the comparison of coenzyme Q bound to mitochondrial membrane proteins among different mammalian species. So if you take the example of mouse, it has low amount of coenzyme Q associated with the mitochondrial membrane, where the cow, it has a higher amount of coenzyme Q in the mitochondrial membrane. Fine. So if there is low amount of coenzyme Q, there is higher amount of peroxide radical. That is what we observe. Can you understand this correlation plot now?
Yes, sir.
So you understand how coenzyme Q, that was ubiquinone, that is important for maintaining the level of reactive oxygen species in the cell. Is that clear to all of you?
Yes, sir.
So if ubiquinone is less in number, then what will happen? Then this superoxide levels will increase. If superoxide levels will increase, there will be Fenton reaction, and the Fenton reaction will cause production of hydroxyl radical, which is very, very damaging to the cell. That's what we have studied. Now let's come back, uh, come to another paper here. So what they have done in this paper is they have inactivated SOD2 enzyme. What does SOD2 enzyme do? What is it? What does it encode? I mean, it is encoded by SOD2 gene. What kind of like superoxide dismutase it is? Copper zinc SOD or MnSOD? Which one?
Copper zinc SOD.
No, MnSOD.
Sorry, MnSOD, sorry.
So it's present in mitochondria, or it's present in the cytoplasm, or is it present in the what were the other option? I forgot about it.
It is present in mitochondria.
Mitochondria. Okay, fine. So in this paper, we will see what happens if you mutate, this enzyme, you inactivate this enzyme, then there is dilated cardiomyopathy. So what is dilated cardiomyopathy? We will see very soon. So how did they mutate this SOD2 gene? So this is the normal SOD2 gene. How many exons? These are the exons. So this is exon 1, 2, 3, 4, and 5. Okay? There are 5 exons. Is that clear?
Yes, sir.
Now, using homologous recombination, they have removed this exon 3 and they have inserted this neomycin resistant gene. Why do you think they did that?
Sir, how they did it?
Homologous clone करके ना, cloning होगा यहाँ पे. They will clone this gene, like there is a vector. Okay, this is something you will read in RDNA class.
सर अभी तक
Yes,
पढ़े
Yes,
नहीं
sir.
ना.
Yes, sir. We know.
NKJ sir will teach you about it. So yeah, I know. It's you know. I mean, you guys have no idea about what is a vector, plasmid, these kind of things.
So we have very
Yes, sir.
basic ideas. So could you please give us some more idea?
So
That
this is essentially a plasmid which has a promoter. ठीक है, एक promoter होता है. Then there is a multiple cloning site. ठीक है, MCS. So this multiple cloning site, they have a lot of restriction sites. Like in this case, it's EcoR1. Okay. So these restriction sites are present. So when you digest this vector using these restriction enzymes, what will happen? It will linearize. And let's say यहाँ पे EcoR1 है और यहाँ पर HIM3 है. So what will happen when you see this in a gel? It will come up like if this is a let's say 300 base pair fragment and this is like 3 kb like total length of the plasmid, then in the gel you will see one band in the 3 kb zone or 2.7 kb zone, usse thoda niche hoga, and in the 300 base pair around somewhere around this, if this is the 100 base pair equivalent, then it will be somewhere here in near to that. So then you will, when you digest it, this is how it will look like. So essentially you digest it, then you, then what happens कि वहां पे the plasmid is linearized. Now you will introduce like this neomycin. You will introduce this clone essentially. The clone is having SOD2. ठीक है. Then SOD2 clone introduced किया, there will be one clone which will have functional SOD2. Another clone. Yes, Tamalika, any question? Questions? I mean, why are you raising hands? Am I seeing it? Is it clear to everybody, or should I repeat
so
something?
what is a clone?
A DNA fragment needs to be cloned in a plasmid so that you can increase the number. Clone essentially is like when you have the same, identical DNA content together, then it is actually called a clone. But we do cloning in molecular biology because we want to amplify our gene of interest. Is that clear?
Yes.
Now, let's say I'll still give you examples. You have a bacterial promoter and you have insulin gene. Okay. So you now transform the bacterial cell. You will insert this plasmid into the bacterial cell. Then what will happen? Insulin will be produced. Yes or no?
Yes, sir.
But ideally, in a normal situation, insulin is not produced in bacteria, right? This is exactly
Yes, sir.
how recombinant insulin is produced. They are not produced in bacteria. Can you tell me why they're not produced in bacteria? We prefer humanized cell lines. not bacteria. Why?
Because the insulin which is produced means after translation it is formed as pro-insulin, which undergoes some other biochemical changes through the Golgi complex and endoplasmic reticulum. From pro-insulin, with the help of a signaling peptide, it forms a pre-insulin and then it is modified to pre-insulin. So since a prokaryotic cell doesn't have Golgi apparatus, so it will not be able to do that
That is true.
processing for which.
Yes, that is true. That is one of the major reasons why that happens. So, the first reason, what you said, is that post-translational modifications, how it happens in eukaryotic cell, is not same as that of a prokaryotic cell. And the other reason is that I think I told about splicing. So splicing does not happen. So in prokaryotes, if you see E. coli, for an example, it does not happen in that case. So that is another reason. So we have anyway diverted a lot from our topic. So what essentially is happening? This is a clone which is having a functional mutate, I mean functional SOD2 gene, and then we are introducing another synthetic I mean insert which has this neomycin. Neomycin is a selectable marker. So do you know what selectable markers are?
Selectable markers are some specific DNA sequences which have to identify a particular plasmid.
Plasmid.
I mean vector. Whatever.
Why,
For example,
why would I need to identify a particular plasmid or vector?
because then I would know the restriction enzyme sites which are present
Let's
with the help of which.
नहीं वैसा नहीं है. Let's say this is the construct that you have created, which has now a mutated neomycin, I mean mutated superoxide dismutase and contains the neomycin gene. So now when you grow this cell in presence of neomycin, this is neomycin resistance basically. This is the neomycin resistance gene. So when you grow this, this if you have cloned it in a bacteria, then it is a bacteria. If you have transfected it into, eukaryotic cell, then in an eukaryotic cell, if you grow it in presence of neomycin, these cells which have this, this construct present, they will be resistant. They will not die.
Yes,
right?
sir.
But the ones which do not have these, they will die. So you cannot expect if you do this, the transfection or transformation efficiency will be 100%. Few cells will take in the DNA, few cells will not take in the DNA. So the ones which will actually take in the DNA, they can be, selected. So that's why it's called selectable markers.
Yes, sir.
Okay, so as you can see, these are the embryonic stem cell clones. Here also, this is like homozygous deletion, heterozygous deletion. So we observe that 16 kb, there is a fragment which is coming up. That might be due to the eco R1, extra site that has been added by eco R1. So this has gone through the eco R1 digestion. And this is the gel what you see. Now SOD2 deficient mice, so if there is homozygous like deficiency, then everything is at 16 kb. So you know what is homozygous and heterozygous?
Yes, sir.
Okay, so this is, you can see here the length of this entire fragment is 24 kb. Only till here is 16 kb. So is that clear? Now why this 16 kb and 24 kb is coming up?
Yes, sir.
Okay. Here in this diagram, you can see wild type and mutant. So this is exon 1, 2, 3, 4, and 5. In the mutant, you only see exon 1, 2, 4, and 5. So one exon is removed. Clear?
Yes, sir.
Fine. This is the MNSOD levels. MNSOD levels we are seeing using Western blot. You know anything about Western blotting?
Not
Yes, sir.
studied yet.
Okay, maybe in the
So-
next class I will bring. हाँ बताओ Mayuk.
So, as far as I've heard, Western blotting involves Western blotting involves the printing. It's like fingerprinting. It involves the staining and work with proteins.
Yeah, protein is correct. So essentially what happens, you run for what is the process of separating a protein? Like agarose gel electrophoresis is for DNA. What happens for protein? What do we do?
SDS page.
HDS phase or polyacrylamide gel electrophoresis. It could be native page also. So you do this to I mean distinguish proteins. So once you do that, then you use a nylon membrane. There are other membranes also. So this membrane, you like put it over the the gel, and this then there are some reactions which are happening. So the protein, it is imprinted onto the membrane. Then you visualize this membrane. So when you see CuZnSOD, I will discuss about this particular process, since you have not read about it, later in the class. I mean, not in today's class, but in the next class. I'll prepare slides for like Western blotting, Northern blotting, and Southern blotting. Yes, Shinjan?
So a DR eta কি স্যার wild type আর mutant?
কোনটা?
So DRE লেখা আছে স্যার D arrow and E arrow. So in the wild type and mutant, that mRNA.
These are essentially primers. So they
আচ্ছা,
are so not very relevant to our context, but they are trying to when sequencing, before doing sequence, you have read about sequencing, heard about it, or is it also new?
no sir, read it.
So forward primer and reverse primer is there. Then you amplify this, you do PCR, and then you send the DNA for sequencing. So to actually understand whether you have actually caused a deletion, or it might be due to something else. I mean, something else might happen. So we confirm it by sequencing. So yeah, as I was telling you, so CuZ and SOD levels, they are there in all the cases, whether it is a homozygous, like in this case, SOD2 is functional. In this case, it is totally non-functional. This is a heterozygous mutant. So MNSOD, it decreases. Can I say that?
Yes, sir.
There is absolutely no MNSOD in this place. There is no band. So it's non-functional. But what about CuZ and SOD? What is the observation that you have?
It is, the levels are constant. The amount is same.
Is it really constant, or it's slightly increasing or something like that? Is it slightly brighter here?
Yes, slightly brighter.
It's like very less actually. This kind of method is not good for quantitative analysis, but essentially what happens, CuZ and SOD levels, they have increased by 25% as reported in the same paper. And that, why do you think that has happened? Any possible explanation? What will happen if SOD2 is mutated and it is non-functional? What will happen? What do you expect to happen?
Reactive oxygen species concentration will increase.
Increase. Okay. What does CuZ and SOD do? What does an SOD do? What does superoxide dismutases do?
It like eliminates the ROS.
Yes. So this is a compensatory increase. Since MnSOD is absent, So I mean, CuZnSOD is trying to compensate for this his absence. I mean, this MnSOD's absence. So that's a compensatory increase. But again, it is not very effective. Okay, it's not like that if you remove one key player, then there are other players who would try to take that place in, but they'll not be very efficient. So let's see what happens to the mice. So this is the SOD knockout, I mean SOD mutated mice. This is negative, and this is the one which has SOD. So what is the difference that you see? What is the first difference?
The size of that SOD present mice is bigger.
If SOD is present, then the mice is having a healthy growth. But if SOD is absent, then the growth is impaired. That is what it appears to be. Now, if you see, this is the cross section of a heart. So this is for the homozygous SOD present, and if the SOD is absent. So what is it? What do you see? What is the observation? RV is right ventricle, LV is left ventricle. I'll maybe give you the observations. So you see that in SOD mutated mice, the left ventricle is enlarged, and the walls of the heart are thinner. Is that correct? Can you guys hear me?
Yes, sir.
I mean, is the observation true? Is it going too much advanced for you people? Is it, I mean, are you people not understanding or understanding? I'm not getting to know this. No answer. Anyway, another thing that we observe that in the skeletal tissues, there is accumulation of lipid droplets in the mutant, I mean mutated or knockout of SOD2 version of mice, whereas the homozygous where SOD2 is present, there is not that much lipid accumulation. Fine. This is another observation. Then we see that here the same observations that we had earlier. Here we see those represented in a graph. So the left ventricle wall and it becomes thinner in case of the mutated SOD2 mutated or inactivated mice. The left ventricular cavity increases. So this essentially, this enlargement of the cavity and thinning of the walls is called dilated cardiomyopathy. Is that clear? So this is exactly what happens. So dilated cardiomyopathy is happening because this mitochondrial superoxide dismutase is non-functional. Lactic acid concentration decreases, and then the amount of ketones they increase. These are the observation. The overall pH decreases. So there is metabolic acidosis. So what is metabolic acidosis? It is the decrease in pH of tissues or cells due to change in metabolic activity. That is what we observed. Even we see that the carbon dioxide concentration in the blood is the PCO2 levels. It gets lower, but the PO2 levels increase. Partial oxygen levels, they increase in case of inactivated SOD2 mice. So what would this result in?
More DNA damage.
More DNA damage, more ROS production.
More
So
ROS production.
it's a cycle, right? Isn't it?
Cooperative process. Sure.
So then they observe the aconitase, succinate dehydrogenase enzymes activity. It decreases in the heart, liver, brain, everywhere. Why do you think that happens?
Because ROS accumulates in every tissue.
And these have the 4Fe, 4S clusters, right? Presence of
Yes, boss.
superoxide radical will cause Fenton reaction. If you remember the slide previous, I mean, we had discussed about it. So what will happen? This will become non-functional by becoming 3Fe4S. So this the enzymatic activity will reduce. We have already discussed about it. So let's talk about the key findings. So homozygous mutant mice die within 10 days of birth. That is, this event is called neonatal fatality. There is dilated cardiomyopathy that we observe. Then there is accumulation of lipids in liver and blood. There is metabolic acidosis, I talked about it, and there is severe reduction in succinate dehydrogenase and aconitase. Now I would ask you one question. Why do the mice survive in the fetus? There is no change in the fetus, but once they are born, after that, like 10 days after they are born, they die. But they don't die when they are in the fetus. Can you think of a reason?
Uh, because of the fact that when it is inside the fetus, the systems are driven by the mother, right? So, I mean, even the exchange of oxygen and carbon dioxide is controlled by the mother. So, the amount of oxygen that is actually going into the fetal, the fetal system is actually being controlled by that of the by the placenta actually, which is gestation. So it might be the fact that after it is born, so that system is not is of no longer use. So the baby is on its own, for which the problem occurs. Means the problem starts after birth. Actually, the problem was there, but it was not.
Any other answer? Antik is the only attentive kid, I assume. I will not assume that, but anyway. Anybody else?
So,
Mayuk? Huh?
so even I am also of the opinion that in the early stage, that is in the fertilization stage, in the embryo stage, the mouse which is born, it receives nutrition, um, developmental factors, growth factors, etc., within the placenta, which is in the mother's womb itself. So it's in that stage where the newborn mouse in the embryo form gets an advantage from the mother itself. Means it's a passive action which actually protects the mouse in the embryo stage.
You all are correct. Any more answers? Anyone else? I'll quickly take an attendance. Anybody else? Okay, let me ask randomly then. Shuprotim, are you here? Not here. Riddhi,
Yes, sir.
what do you think? Have you heard the question?
So I'm not sure.
Okay. Not sure regarding what? Regarding the question or the answer?
The answer.
Okay, that is fine. But I would like to ask you, how is the course? How are you finding it?
So it helps that the videos are available. So, like the last lecture, I was looking at it before this class. So when I'm able to go at my own pace, it helps a bit more.
So have you been taught like this before?
Yes, so during COVID, this is how we were
No, no, no,
doing
not
classes.
not in online mode. I mean, in this way, like we are discussing about research articles.
No, so we don't really discuss articles much in class. We have a syllabus that we follow.
That I know, that I know. But very soon, when you will be going to your higher studies, assuming that you will do it in the same field, you will be getting a lot of research articles to read from. And one thing that I personally felt that I lacked in my BTech was there was nobody who actually taught us from it. Nobody who told us that how are these things interpreted. So what I'm trying to do here is essentially for you people to uh, just read these things and try to understand. I mean, I'm definitely sure most of you are not maybe that good at it at this particular stage, but it's a starting point. You have to start at some point. So it's better if you start now rather than later. So, okay. Thank you, Riddhi, for your opinions. Anybody else?
Sir, can you please explain once more that dilated cardiac myopathy?
Which one? Which part?
The previous slide.
This one?
The previous. Yes, sir. This ES clones. What is this ES clone? What is ES?
Embryonic stem cell.
That's embryonic stem cell. Okay. So, sir, can you please explain once more that why this band is not visible in SOD2 mice? This part I couldn't hear because of network problems.
So see, this is the actual, I mean, how to remove the annotations. I don't know. Wait, just wait, I see. Yeah, it's getting removed. This is the functional SOD2 gene, right? This is the mutated one. Is that clear?
Yes, sir.
Okay. So there are two EcoR1 sites. And the length of this, the length between these two EcoR1 sites is 24 kilobase pair.
Yes, sir.
Now, when I introduce this neomycin, it also comes with a EcoR1 site. So now when I digest the same, this mutated like DNA with EcoR1, it will produce a band of 16 kilobases. Clear?
Yes, sir.
Now, when all the cells, they have this version of, DNA, then they have only the 24 kilobase pair band. There is no 16 kilobase pair band. Clear?
Yes, sir.
But now when I introduce this mutant, let's say it's in the heterozygous form. So when it is in the heterozygous form, then both the bands are present, 24 kilobases and 16 kilobases. It makes sense.
Yes, sir.
Now when only these mutated, only this one is present, only the mutated SOD2 gene
So,
is present, then there is only 16 kilobase per band, no 24 kilobase per band.
okay, understood.
Okay, so any other answer to why do you think that nothing happens in the fetus? Anybody not from BT? Everybody is from BT. Shreya, Shreya Nag, Shreya Basu. We have two Shreyas here. Any one of you? People are, I think, reliant on the recordings. Shagnik, not here. Fine. Well,
So can
so
you once repeat the question?
question. Okay, the question was that that homozygous mutant mice, mutants of SOD2, they die within 10 days of birth. But why don't they
Yes,
die
sir.
before birth? Why don't they die when they are in the fetal stage? That was the question. Anything pertinent to today's class? Maybe metabolic rate. I've given you a hint. Anyone would like to
So
attempt?
you said that lesser the metabolic rate, more is the lifespan of a particular organism. So within a
Mhm.
fetus, the metabolic rate of a particular organism is almost nil. Like
Right, right,
everything is done by the carrier. So
right.
in that reason, for that reason, sir, the oxygen stress is not at that much,
That is
and
exactly the answer.
that's why the organs are not having the developmental defects because
That
of
is true.
extra protection,
That is
external
true. That
protection.
is true. That is exactly the answer. So the metabolic rate of a fetus is very low. Because of this, the extent of DNA damage is very low in the fetal stage. But once they are born, this story is different. So can you people now, I mean, read research papers and make observations and inferences?
Yes, sir.
Because it will be given. I mean, in the authors will write everything there. So I will be giving you an assignment based on like these research papers only. And even your presentation will be like this. So essentially you have to present something like this. Only one paper, not multiple papers. So you'll all be given like in a group of two, maybe you'll be given one paper and you have to present that paper. So that will essentially, this entire course is designed so that you people know how our research papers read and how to derive, the important results out of it, how to, give the story. So that is all for today. And any more questions? Do you need the research papers to be given to you, or you want to find it out yourself?
Sir, if you please give us at least the DOIs, it will be good, I guess.
I will upload the entire for the first half of the course. I'll upload the entire, I mean, all the papers that I've referred to.
Okay, sir. Okay, sir.
I'll upload it today itself. Fine. So is that clear? I mean, is it
Yes,
okay
So
with
can
all?
you upload
because
the PPT as well?
PPTs are uploaded, no?
So I could only find the video lecture. I couldn't find the PPT.
Is it the same for everybody? Like nobody is responding. How would I
in the
know?
Teams app, there is only the video.
No,
There is
no,
only the video.
don't search for the Teams app. I think I gave you a Google Drive link. Wait,
Yes,
I'll do
sir.
it
That
right
means
here. I'll do it right here. Then it is better because I don't know, you people will not respond otherwise. Just give me a moment. I'm sharing it in the group. All of you are in the group, the WhatsApp group.
yes, sir.
No, who said no?
So we didn't got any Google Drive link. We only got that Excel sheet thing. That's it.
Yes, so you
Ah,
only
to
got the.
open the Excel sheet.
So there we can see only the attendance, like who are joining.
हाँ उसके पास लिंक नहीं है क्या? Timings, then study material. See,
उतना नहीं देखना था.
I really don't know what to say. So please go through it. I'll also upload the research articles on your request. I'll do that. Anything else that you would like to say pertinent to this lecture or maybe previous lecture or something maybe out of this course? Anything?
Sir, in the point 3, it is written the accumulation of lipid in liver and blood. So, how does that occur with respect to this mutated SOD2 gene?
That is what they observed. Like so, I talked about cardiomyopathy in the first lecture. Also, I think I also talked about arteriosclerosis. If you remember,
Yes, sir.
there is accumulation of lipid with age. So maybe something related to that is happening. This is how you find out the gaps in research. So
Yes, sir.
you definitely found a gap. So I don't know whether the people have worked in this aspect, but probably they have. I'm not sure. Wait, I'll just stop the recording here for the sake of everybody. How to stop that? Just give me a moment. Any more questions?